Okay, welcome everyone to another SOL seminar online. Today we are glad to have here Andrea Arrones. She is working on the Institute of Conservation and Improvement of the Valencian Agrodiversity at Polytechnic University of Valencia in Spain. And she is a Bachelor of Science in Biotechnology and Master of Science in Plant Breeding. Currently, she is working on her PhD on the Solanaceae breeding group, and her thesis is titled Development and Exploitation of MAGIC Experimental Populations in Eggplant and Tomato Genetics and Breeding. She's focused on the development of mapping populations such as different eggplant, RILs populations, the first eggplant MAGIC population, and one tomato MAGIC population. So today, her talk is going to be about the dawn of the age of MAGIC populations, a case of eggplant. And with this, I thank Andrea for being here and show us her amazing work.
Thank you very much. So I'm going to share my screen. Here. Okay. So hello, everybody. Hope everything is going well and you can see my screen and the red dot because I'm going to point out some things. And first of all, thank you. Thanks, Rocío. And thank you for giving me the opportunity to be here today and to participate in these seminars. And I'm going to introduce myself a little bit and also my group of research. So as I said, Rocío, I'm a PhD student of the Polytechnic University of Valencia in Spain. And I'm working in the Solanaceous breeding group of the COMAP Institute. And in our laboratory, our main line of research is eggplant, but we are also working with tomato. So we are mainly specialized in the development of experimental, interspecific experimental populations. And for instance, we are working in different introductions lines, combining cultivated tomato with wild species, such as Solanum incanum, Solanum insanum, Dasyphilum, and Eleghnifolium. And these mapping populations are being used for QTL mapping and candidate genes identification for different traits of interest. So during my PhD, I've been working in different development of two MAGIC populations. One
Andrea,
in eggplant.
Yes. We cannot see your slides.
No?
Oh, no. Yeah, we can see the slides. Maybe some problem with your Zoom, Pietro?
You can see the slides. Okay, sorry. Okay.
Okay, so you can see it? Yes?
Yes.
Okay, thank you. I'm sorry. So during my PhD, I've been working in the development of two MAGIC populations, the one in eggplant, which is the one I'm going to present here today. But I've also been involved in the development of a tomato MAGIC population, which I'm going to introduce briefly at the end of the presentation. Okay. So with this work, I would like not only to introduce you in the MAGIC world, but also to show you the potential of this kind of populations. So MAGIC populations or multi-parent advanced intercross populations are panels of recombinant inbred lines, which are genetic mosaics of a combination of multiple founders. One of the main important things to take into account before initiating the development of a MAGIC population is the selection of the best founders, since they should cover a genetic, geographic, and phenotypic diversity to exploit the potential of the MAGIC populations. So it is also important that these founders are well characterized at the molecular and physiological levels to produce practical results, because it will be crucial, for example, for haplotype prediction or for causal variant identification. So once you have selected your highly diverse founders, they are pairwise intercrossed to produce the F1 hybrids. And they are subsequently intercrossed in pairs to obtain the double hybrids. Then in order to achieve a complete admixture of all founder genomes, the double hybrids are intercrossed following which is called the chain pollination scheme, which each individual is used as a female parent, but also as a male parent for the following plant to finally generate the quadruple hybrids. Finally, to finish this magic population development, you will need to generate or develop different generations of selfings to generate the final immortal fixed lines with a high degree of homozygosis. To do that, these immortal fixed lines could be generated by 5 to 8 generations of selfings through single seed descent, or they can also be generated by double haploid production. It is true that this way, the double haploid production is the fastest or the shortest way to produce fully homozygous individuals. However, the additional rounds of selfing through single seed descent can introduce novel recombination events, which is what we are looking for in this kind of populations. So, in addition, not for many crops, there is an optimal double haploid protocol established because this technique is very strongly dependent on the species and the genotype. And for instance, in eggplant, the generation of double haploid productions is still so tricky. So with few exceptions, this way, the single seed descent approach is the most used for the magic populations, which are published until now. So once you have seen how magic populations are, the question is why magic populations? So although they present some disadvantages, mainly related to their development because they have very difficult and long time to be developed, they overcome the main limitations of bi-parental populations. So as I have said, the clearly first disadvantage of this kind of populations is that they required huge investment in time to be developed due to their difficult scheme involving lots of intercrossing between the multiple founders, but also the selfing generations to achieve the homozygous individuals. So we have already talked about the production of double haploids, which could be an option. But another option we propose here is the so-called speed breeding approach, which main objective is to accelerate breeding and shorten generation time by growing plants in under controlled environments or with controlled photoperiod and temperature regimes. So actually there are speed breeding protocols have been published recently in different crops and seem to be very, very efficient. So we also propose this approach for accelerate the development of magic populations. As I have said before, another disadvantage or drawback of magic populations is the complexity of the crossing scheme, which could also present problems during population development. However, the combination of the multiple founders enrich populations with higher genetic and phenotypic diversity when compared to those populations coming from bi-parental crosses. So in addition, the multiple cycles of parental intercross result in an increased level of fragmentation and recombination rate, which is going to increase the power for QTL detection. And finally, another disadvantage of these populations is the large population size that is required for exploit the power of magic populations, which difficult the development, but also difficult or limits the number of phenotypic traits that you are able to evaluate. So the most important thing is to clearly define the objectives or the goals before initiating the magic development and take into account all of these limitations. So now you know what's a magic population and their advantages and disadvantages. I'm going to present you the eggplant magic population we developed in the group to show you the potential of these populations for QTL mapping and candidate genes identification. So we developed the first interspecific eggplant MAGIC population, selecting 7 Solanum melongena accessions as founders, but also 1 wild Solanum insanum. And this Solanum insanum founder was selected since it is known to be a source of phenolic compounds, but also tolerant to some biotic and abiotic stresses. And as in the final population, it is expected to achieve a 12.5% of the wild genome. It could be very interesting for the study of different traits given by this wild germplasm. So introduction of the wild species as founder is an interesting option to broaden, to further broaden this genetic and phenotypic diversity of the population. However, it's very challenging to introduce these wild species because some difficulties could be associated to the crossing design, such as linkage drag or partial sterility of the hybrids, for example, among others. So in our population, the introduction of the wild founder led to the generation of some individuals carrying the wild cytoplasmic background, since the Solanum incanum founder was used as a female parent in the intercross. And this could be very interesting for studying some traits. So once we had the F1 hybrids, we generate the double hybrids, as I've said before, by following a chain pollination scheme, and then to obtain the quadruple hybrids, and then to accelerate these generations of selfings, we grow plants in small pots, which allow us to grow high number of plants in a reduced space. And selfings were stimulated by mechanical vibration. Based on the results we obtained here, we are now working in a speed breeding protocol for eggplant. And we have now some trials in the greenhouses testing different growing conditions, different pot size and different watering conditions. So we will see the results. We can get something interesting to develop a speed breeding protocol in eggplant. So once we have the population, okay, so as an intermediate step of the final population, we check how the population was going on, and we generate 420 individuals from the 3rd generation of selfing. And these individuals, to see how the MAGIC population was going on, we genotype them by high throughput genotyping using the 5,000 probe SPET platform specific for eggplant. This platform was developed by our group in collaboration with another Italian research groups. And this is an amplicon sequencing strategy which is able to identify lot of SNPs, large number of SNPs by low rise. So after the genotyping, as a result, it was observed that the S3 individuals exhibit low heterozygosity levels of about 6.87% on average. So this value is very similar to an F5 like bi-parental intercross generation. So we were very advanced in our, or at least the selfings were going very well. So we also perform a principal component analysis in which we include the genotyping data for the founder lines and the F1 hybrids. And as you can see, the plot makes sense since hybrids are in intermediate positions of the founders. For example, here you have a D founder and C founder, and you have here the F1 hybrid, or here closer B founder, A founder, and you have here the F1 hybrid. We also observed that there is no population structure in our population, which is also an advantage of MAGIC populations against natural germplasm collections. And there's also no differentiation between cultivated or wild germplasm, since no clear clustering was observed here, you can see. This population was also phenotyped, and genome-wide association study was performed for fruit peel pigmentation. As you can see here in this picture, eggplant is characterized by a great diversity of colors in its commercial maturity as a result of the combination of two main pigments, which are chlorophylls which are giving this green color or this green color, and anthocyanins, which are giving this purple color to the eggplant fruits. And in this study, we aim to elucidate the genetic control of these two pigments. So as a result of the GWAS developed for the anthocyanins presence in the fruit peel, in the Manhattan plot, we identify a very significant association peak at the end of chromosome 10, under which we identified the MIB113 gene as the best candidate, and it was previously described as a regulatory transcription factor controlling anthocyanin synthesis in eggplant. We identified different variants which were predicted as high impact effect on the protein function for the MAGIC founders that do not present a fruit anthocyanin in the peel. Specifically for founders A and C, we identify which as you can see here, they are green fruit. They did not present anthocyanins in the peel. For founders A and C, we identify single frame sheet variant in two different positions, and we also identify a disruptive selection for founder F, which is white fruit. So we identify finally 3 different mutations which explain the absence of fruit anthocyanins in all the individuals for our population, coming from these mutations found in the 3 founders without anthocyanins in the fruit peel. Then we also develop a GWAS analysis for the presence of chlorophylls in the fruit peel. And in the Manhattan plot, we identify a very significant association peak at the end of chromosome 8, under which we identify the gene APRR2 as the best candidate, since it was also previously described as the pigment accumulation and chloroplast development promoter in another solanaceous and cucurbitaceous crops. We also identified contrasting mutations between accessions with and without chlorophylls, and in particular, in our MAGIC founders, we identified 2 small deletions which result in a change in the ring and frame that did not, these 2 founders did not present chlorophylls in the fruit peel. But for people who is not working with eggplant, we have now available 4 different eggplant reference genomes, and 2 of the 4 reference genomes are developed from accessions without fruit chlorophylls. And in these ones, which have no chlorophylls in the fruit peel, we also identified a large deletion at the end of the gene, which was validated experimentally by a PCR amplification. And as a result, we identified 2 different mutations in this gene for our MAGIC populations, and another more mutation in the eggplant reference genomes for this gene, which explain the absence of chlorophylls in the fruit peel. Going back to the GWAS in the Manhattan plot results, a minor but also significant peak was observed at the beginning of chromosome 4. Under this peak at the beginning, no candidate gene with contrasting mutations for the different phenotypes was identified. However, previous studies associate this exactly same region to another trait also related with the chlorophylls presence, which is the netting trait. So as a break, to understand the implication of this trait in the appearance of the fruit of the eggplant fruits, as you can see, the distribution of chlorophylls in the fruit could be uniform all along the fruit, or could appear like irregular pattern, which is the so-called netting, and which is also known as reticulation or variegation. This trait, it's more commonly present in the wild species, and it's difficult to find in the cultivated species or commercial varieties. However, it can be interesting to introduce this trait in breeding programs, because in combination with chlorophylls or anthocyanins broadens the color palette of the eggplant fruits. since the MAGIC population was phenotyped only for the presence or absence of chlorophylls, without discriminating between the distribution of chlorophylls, we determined that there was a possible relationship between this association peak at the beginning of chromosome 4 and the netting. So as we suspected that, we decided to further investigate this trait. So previously studies which were working on the netting trait demonstrate that it was controlled by a single dominant gene following a 3-1 segregation in F2 populations. Indeed, we proved that because we developed an F2 population between a white Solanum melongena and a wild species Solanum insanum, as the donor of the netting trait. And this population was subsequently used for BSA-seq analysis that allow us to reconfirm the candidate region previously proposed in the GWAS of the MAGIC population, because we also identified a peak of significance at the beginning of chromosome 4. To reduce the number of candidate genes annotated under this genomic region, we used advanced backcrosses from intermediate development stages of 2 different populations of integration lines that we were developing in parallel in the laboratory, and which were segregating for the trait one more time by the contribution of the wild founders, Solanum dasyphilum and Solanum insanum. The individuals or the advanced backcrosses coming from these 2 populations, which present netting, were genotyped and were self-fertilized for fine mapping. And we not only reconfined the candidate region on chromosome 4, but we also reduced the genomic region to one with only 14 genes annotated in the reference genomes. And among these 14 genes, we select the GLK2 gene as the best candidate, since this was also previously described as a regulator of chloroplast synthesis in other species. As I mentioned before, it's true that we didn't find at the beginning contrasting mutations for the 2 contrasting phenotypes. However, we identify in the intronic region a mutation that was shared by all the netting, the individuals presenting netting. So we started to look for this region, and by studying the different reference genome, I said before, and synteny with tomato, and also some transcriptomic data, we identified or we determined an erroneous annotation of the GLK2 gene in the available eggplant reference genome. As I said, as I showed you before, the reference genomes, if you remember the fruits, these fruits did not present netting. So we can understand that the annotation in these genomes is erroneous. So we proposed, or we suggest the existence of an additional exon, where a small deletion was identified that give rise to a premature stop codon, which disrupt the protein sequence and explain the absence of netting in our population. So to understand the differences between uniform and irregular pigmentation of chlorophylls at the chloroplast level, we analyzed different green pill tissues by confocal microscopy. And here we took fruit peels from different samples, from samples with netting and samples without netting, with a uniform distribution of chlorophylls. And as you can see, we observe more developed chloroplast with higher chlorophyll accumulation in these pigments or in these samples or tissues coming from netted samples. This could be the reason why the green color coming from the netting persists and remains during ripening until it turns to yellow when the fruit is completely mature, while the chlorophylls coming from the uniform pigmentation fed more quickly, giving rise to this yellow pigment during the ripening. So here, all the results that I've presented till now were validated on a set of approximately 280 accessions from the eggplant core collection developed under the G2P project. And not only we validate our results, but we also identify new variants in the proposed candidate genes, which indicates that these genes or these traits have been arisen and selected several times and independently throughout the domestication process. For example, in the case of the APRR2 gene, which is the responsible for the uniform chlorophyll pigmentation, we observed that the different gene variants display different and specific geographical distribution associated with a specific phenotype. And this might be related to the domestication flows of a plant together with local preferences. So all these results I've shown you until now are the already published results, but we have continued to work with the MAGIC population, the eggplant MAGIC population, and we have now finished the population with the 5th generation of selfing. These final 326 S5 individuals have been genotyped by scheme whole genome resequencing, and they have also been phenotyped for more than 60 traits in 2 different environments, here in Valencia, but also in Almeria in Spain, where it is produced the majority of the eggplant which is commercialized in Europe. And as I have said at the beginning of the talk, we have also developed an interspecific tomato MAGIC population combining Solanum pimpillenifolium and Solanum lycopersicum cerasiform accessions, which are the ancestor and the closest relative to cultivated tomato respectively. And these 8 founders were selected due to they were representative of the genetic diversity and geographical distribution of these 2 taxa. And they cover a broad a broad range of phenotypic diversity, including plant and inflorescence architecture, also leaves, fruits, flowers, and fruit traits. So after 5 generations of selfing, a total of 354 S5 individuals were generated, and they have been genotyped by the 12,000 probes tomato SPET platform. And the preliminary results reveal low heterozygosity levels of these final individuals and absence of population structure once more time. As you can see in the principal component analysis, we also include the genotyping data of the founders. And as you can see, all the Solanum cerasiform grouped in the negative side of the principal component analysis, while the Solanum pimpillenifolium accessions grouped in the positive side of the first principal component analysis. And here you have the 2 Solanum lycopersicum cerasiform accessions, which are the closest to the cultivated tomato, so they are here in the corner, in the left corner, which makes sense because they are far away from the other founders. In addition, we are phenotyping this tomato population also for a lot of traits, and now there are several trials which are currently under development, including plant and fruit traits, disease resistance, phenylpropanoids accumulation, and also nitrogen use efficiency, among other traits. So with that, I've shown you all the work I've performed during my PhD thesis, which I'm going to present in a few months. Hope everything is going well. And finally, I would like to thank you for your attention and for the opportunity to give this talk, but also to my advisors, Jaime Proens, Pietro, Santi, and Mariola, and also for all the PhD students in the lab and postdocs, but also for our collaborators from the G2 PISOL project, which are Maria Jose here from the COMAB, Giovanni from the NEA, Lorenzo and Ezio from the SAFA in Italy. Thank you very much.
Thank you, Andrea, for such a great talk and all this amazing work. It was really, really nice. So now we are open for questions. You can just raise your hand, or you can actually write a question and I give you the word. As well, you can write your question and I will say for you. In the meantime, I do have a question, but it's mostly about future work. That are you thinking about just checking all these different breeding systems and everything in other species or some wild species related?
Yes, here in the COMAB we have a seed bank, so we have the availability to work with different species. So we always try to check our result in different species because we are, as I have said, in eggplant, for example, we are working with different wild species. So we try to check it in all the species because we have to sequence the genome. So yes, for instance, for the netting trait, all the wild species show this trait, and we check it in our species, and it was like this. So yes, we have the opportunity to work with a lot of species, so we are trying to use them.
That's great. So that will be a future postdoc, or yeah.
Yes, I'm looking for. First of all, I want to finish my PhD and then look for future opportunities.
That's great. Thanks.
Thank you.
Anyone with questions? Everyone's a bit shy today.
Yes.
Luke, do you have a question?
No, I was going to ask something similar to what you already asked, so it's been answered.
Okay.
I would like to make a comment, not regarding the presentation of Andrea, which I know quite well. It's for you, Rocío, because the timetable that you have sent in the email, you should review it, because it says that for Europe, for GMT+2, it was at 5, and
yes.
Here it was at 4. So maybe
yes,
some people
sorry.
Yeah.
Actually, Thibaut mentioned that at the beginning that there were this mistake because there was a time change in Europe as well that we missed. So we are changing that timetable for next webinar.
Very good.
I'm sorry, and thank you for for mentioning it. Yeah. Okay. No other question. In that case, we are ending this seminar. So thank you very much, Andrea. That was a great talk.
Thank you.
And of course, like, you can contact Andrea with questions, or you can comment as well. This video is going to be available in YouTube and in our webpage. And next talk is going to be presented by Pilar Zamora Tavares from Mexico, mostly about physaloids, and hopefully I think she's going to present about the new genus of physalid cataract that was recently published. So we are, like, next talk is going to be December 1st. Well, I hope to see you all here. So thank you.
Thank you very much.
Bye.